This is an A-level biology presentation looking at sampling. We're going to be looking at a number of different concepts as we go through this section. We're going to look at different ways in which we can do sampling. We're going to be looking at bias and how to remove bias. We're going to make sure that we look through the mark capture release method and the different ways we can use the mark capture release method. So first off, get started, is there's a picture on the left hand side. All you have to do is identify what does that picture show and how do you use it. If you pause the video now, come up with an answer, and then you can restart the presentation. So that is a point quadrat. So don't always assume that a quadrat is going to be a square. And how you use it is basically each of the needles goes in, or each of the pin goes in, and any plant that you're investigating that touches those particular needles are counted in your population size. So it's a slightly different variation of what you would know as a standard quadrat. Now there is an extension question. It says in sampling, is it better to have lots of samples or a small number of large samples? Well, the answer is indeed a lot of small samples. The more samples you have, the more accurate your data, but there does come a point where your sample size is so small that it's not actually representative of the population, so you need to make sure that your sample size is appropriate. So sampling one person out of every 5 million is not what you'd call a appropriate sample size. There are four pieces of equipment in front of you. We've already mentioned one, which is of course the point quadrat. You should be aware of the other three from GCSE. Again, spend a minute, pause the video if you need to try and come up with the names of each of those pieces of equipment. The top left picture shows what is called a pooter. Note the little bit of mesh over the end of the straw that you suck on. Every so often someone decided not to put those on, and quite regularly you would have little bugs being sucked up the second straw and into the person's mouth, which they only made that mistake once. In the middle you have the more traditional quadrat, and then on the right hand side we have what's called a sweep net. Give an example of random sampling. So pause the video and think of random sampling, and you can also go on and have a think about what is systematic sample. So give an example of random sampling. When you've got your example, you can restart the presentation. Well, in terms of random sampling, the thing that everybody thinks of, I'm hoping you thought of, is standing in one place with a quadrat and throwing it. Well, unfortunately, folks, despite what you may think, that is not random sampling. The reason it's not random sampling is because there's only so far you can throw. You will only throw in one direction, so anything to beside or behind you, if you're not throwing that way, can never be sampled. And the place you're standing on could also not be included in the sample unless you have a particular interest in hitting yourself with a quadrat. Random sampling only occurs when every part of the particular area you're sampling, or every organism in the particular population you're sampling, is equally likely to be chosen. So you can't have any bias, you can't have a preference for any particular type of organism or any particular area. So in the classic example for a quadrat, we have bias. What we should do is we should create a map of the area we want to sample, split it up into grids. We then do exactly the same in the actual area using ropes and pegs, split it up into grids, and then we allocate each grid a number and literally draw bits of paper out of a hat, and that is how we get our random sampling. Okay, standing in one spot and throwing is not random. Don't say that for A-level, folks. Everything has to be equally likely. As I said, the maps, gridding out the map, gridding out the actual area where you're taking the sample from, and then allocating numbers to each bit of the grid and drawing bits of paper out of a hat is truly random. We can also do what's called systematic sampling, and systematic sampling is taking regular samples often along a transect. So you take a particular line of travel across an area to see how a population changes as you move along that area, and normally you're moving away from or towards a particular phenomena. For example, you can move up along a beach away from where the water's edge is, or you can move away from, say, a tree in the center of a field and move further away from the tree and see how the population changes too. Remember, we have to make sure that we're talking about the number of organisms is the frequency, or when we're using quadrats, we can talk about percentage cover, and you got to make sure you're using the right one in the right time. For things like grass, moss, and so forth, the individual plants are so small, you wouldn't talk about the frequency of them, you would talk about the percentage cover. So what percentage of the quadrat was covered with that particular species. However, if you're looking at something like a flowering plant or something slightly bigger, you may move on to frequency, and frequency is how many individual organisms there are of that particular species inside the area. So it depends on what exactly you're sampling if you use frequency or percentage cover. So our previous method using quadrats is normally used with plant life. When it comes to animal life, we normally use the mark capture release method. In a mark capture release method, there's several steps. You should have covered at GCSE, so pause the presentation, have a think of the steps you would have taken in order to do the mark recapture method, and then you can restart the presentation whenever you're ready and think you've got all the steps. First question, did you remember that you have to first capture them, or did you start by marking them? It's a bit that everyone seems to get wrong at A level. They talk about marking stuff, releasing stuff, and recapturing stuff, but they forget about that first capture. You've got to capture them first off before you can mark them, and then you release them, and then you do your second capture. So for that reason, I refer to it as the capture, mark, and recapture method. Mark and recapture method is obviously the name, but the capture, mark, recapture method just keeps that first step in your head. So the steps that you would do is you'd capture them, count them, mark them, release them, recapture, and count. Now there is a related equation. Again, folks, you need to memorize this equation off by heart. What you have is you get the number of organisms in the first sample, you multiply that by the number of organisms in the second sample, and then you divide that number by the number of organisms in the second sample that have the mark, meaning they were included in the first one. And that'll give you the estimated population size. So this is an example of the kind of question you could get in the exam. You're given a table, you're asked to calculate the population size for organism C, pause the video, have a go at the question, and then when you've got your answer, we can go through it. So pause the video now. So the question asked you to calculate the population size for organism C. The first thing you need to be able to do is actually recall the equation that you need to use. Then you take the information from the table and put it into the equation. So the first sample, it says in the table, the number of organisms in the first sample for organism C is 4. Now the second sample has been a little bit tricky here. You need to look carefully to see that the second sample has actually been split into those that were marked and those that were not marked. So to get the total second sample, you actually need to get the 12 that were unmarked and add the 2 that were marked. So the population for the size for the second sample is actually 14. And then we've got to divide that by the number in the second sample that were marked, and that tells us from the table that there were 2 organisms. So our calculation, when we put the data in, we should have 4 times 14, and then divide that by 2, which is 56 divided by 2, which will give us an answer of 28. So as I call it, the capture, mark, and recapture method has some assumptions associated with it, and these assumptions we have to take into consideration when we're looking at real life data. The 5 pictures represent 5 of the assumptions. What I'd like you to do is pause the presentation and see if you can work out what each of these different pictures represents in terms of assumptions when using this particular method of sampling. Pause the presentation now and restart it when you've got your answers. Well, the answers are, in the 5 different ways, the first picture represents the fact that the proportions in the sample must be representative of the actual population. So you can't have a sample that's disproportional to the population. It has to be representative. We also have to assume that any of the organisms that get marked and released for the first time, they have to evenly spread out. There can't be any particular bias, they can't run in any particular direction. They have to randomly spread out in all directions. Third one is that we've no emigration or immigration. Number 4 is my favorite. Number 4, of course, is that the markings do not give any sort of disadvantage to the organism. We can't have markings that make the organism disadvantaged. One of the classic examples is where there was markings put onto animals and it was paint, but it was paint that only showed up in certain wavelengths of light. And the problem was that the predator of this particular organism was actually able to see in that particular wavelength of light. So the markings stood out to that particular organism's predator. And as a consequence, it obviously had a huge impact on the data collection until somebody noticed. So you got to make sure that the markings do not disadvantage the organism. And lastly, of course, we have to make sure the markings can't be rubbed off or lost. There's no point in having something that's water soluble if we're marking organisms that spend half their time in water. So you've got to make sure that whatever you mark them with is appropriate and doesn't get rubbed off or lost. Well, with this new information, we can actually update our description of the particular method because we can add in a few extra statements. So when we get marking, we can now mark in a way that does not disadvantage the organism. And when we release, we have to allow time for the organisms to disperse randomly. They have to be allowed time to spread out across the entire area that we are sampling. We can't just release them in 10 minutes later to try and catch them again. If they're all going to be still there, it will give us obviously the wrong sample. And so just to finish off, thinking of all the content that you've gone through in the presentation, the terminology and all the different concepts, and make a list of what would the exam question be? What kind of things would they ask you in the exam based on the information that you've covered? And then go back and see, once you've got your list of questions, can you actually come up with an answer to each of those questions?